Thank you, Jennifer, for sitting down with me.
Thank you for interviewing me.
Well, we're here at AgSmart 2023. This is the first time the Field Crop Development Center Field Day has taken place as part of AgSmart. It's also the 50th anniversary of the FCDC. FCDC has been a part of Olds College now for about 3 years, so lots of milestones. For you, what's the biggest benefit of the field day taking place as part of AgSmart?
Well, probably one of the more obvious reasons would be just the number of people and the size of this field day here at AgSmart versus the smaller one that we used to have, which was more specific to seed and our breeding program. But, here we get a diversity of people coming from more areas, from, producers, farmers, seed growers, as well as other people within the industry. And as well, we're at a college, so we have students that come in. And it's always fun to be able to talk to students that are very interested in my work. And it gives me an opportunity to show off what I do in my lab.
Well, speaking of your work, what are you talking about here today?
Well, I'm the lead in the Biotech lab at the Field Crop Development Center. And what I'm talking about are some of the tools that we use in the lab to help the breeders better select traits in their lines, the breeding lines. This is going through this pipeline to finally get, the perfect variety at the end. And if you look at a traditional breeding program, the breeders would cross the plants and then grow plots in the field and then look at them and try to make a decision. But not all the traits that we're interested in putting into these new varieties are necessarily easy to see if we look at them. So if you're looking at quality, malt quality, feed, forage quality, it's going to require a wet chemistry. So you can't see it by looking at the plant for sure, right? And then there's some other areas that are problems when we're looking at disease resistance. So depending on the genetics behind the resistance, we may want to actually look at the DNA, and the same for the quality. We can look at the DNA and we can come up with, it's a relatively quick method we can do in the lab. And by looking at that DNA, we can predict how it will perform in the field. Does it have that disease resistance? Does it have that quality?
What are some examples of recent advancements in this area that you can point to say, this is how it works, this is some of the benefits of it?
So the one thing that we've seen probably over the last 20 years, quite often people, when they ask me about doing molecular markers or looking at the DNA, they think it's going to be really expensive. But because of the developments over the last 20 years, we have cell phones now. All that technology has come to the lab. We've also developed new techniques that require less DNA to do the analysis. And we also have these new techniques where we can actually use fluorescent dyes. So we put it in a special type of equipment. It will do the analysis on the DNA, and then depending on what, and I haven't defined yet the markers, but what marker is there, it will give us a different color and record everything, and it makes it super easy. We don't have to go through it as an individual and score everything onto a specific table. So, the cost has gone down. It's less labor and it's more accurate. And we can take more data in now too. So we can start taking digital data, from drones and fields, and use that as well to help us come up with better markers and better predictions for the farm or for the breeders, and then ultimately have a better variety for the farmers.
Well, one of the things we're talking about here today is sustainability. It's a huge topic right now. And how does all this ultimately contribute to better sustainability for the seed industry, for example?
Well, we have several projects actually that we're working on to get more funding, and one we just recently received or potentially will have. It's not 100% yet, but it looks good. But what we want to do, and sustainability for farmers is not new. I think farmers have always been interested in sustainability. Nobody wants to use more nitrogen than they need to. And our breeding program, actually, about 15 years ago, we started looking at nitrogen use efficiency in our breeding program. And our current barley breeder actually was hired on that project. And from that initial work that we've done already in sustainability and nitrogen use efficiency is we have a new variety, AB Standwell, and that is 6% better for nitrogen use efficiency. So that sounds like a small amount, but the problem we have, not every trait that we want to transfer in is controlled by 1 or 2 or 3 genes. Quite a few of the ones that we're really interested in doing, like increasing yield, increasing forage yield, increasing nitrogen use efficiency, FHB is a disease like that. It's many genes and they have small effect. So this goes back to the project sustainability, and this is an area that we're moving into in addition to the marker-assisted selection that we currently use. It's called genomic selection. So instead of just focusing on specific regions of the genome, we're focusing on the entire genome and using that information plus information from the field and from the plots. It can be done what we call phenotyping, the way the plant looks, digital data from the yields, and which can be collected through drones and other types of remote sensing. We're taking all that data, combining it together with the genetic data, and we have new software and quantitative genetic analysis methods. We can build algorithms, and probably in the future we may even start to see more AI. So using AI methods and programs to help the breeders make better decisions on these traits that are very complex. So, and this is sort of where some of our new research and our projects are going. It's a big area, so we're collaborating with other post-secondary institutions. We're looking to collaborate more with other research groups such as AFSC, but that's a very large project, but it's definitely capable and a direction we can go to as we see our ability to be able to handle more and more data.
It's kind of over my head, but it'll be interesting. This is the 50th anniversary of FCDC. So in another 50 years where things might be, say 50 years from now, it must be kind of mind-boggling to think about that.
Yeah, I think, the advantages of some of these techniques is we can get rid of the stuff that's never going to be good enough, right, earlier on. And so, I might suggest, maybe we can use AR, we're doing genomic selections, we can do marker-assisted selection early in the breeding process. But what that does is it helps us reduce the number of lines that have to go through that pipeline before we realize they're no good. So if we can get rid of them earlier, then we can do more crosses. And this is really important for the farmers. So, with the changing climate, more extremes, if we discover there's a gene in some germplasm, in Africa or, that we can bring into our lines and we want to do it quickly, it can be a problem because what we want is those traits to be brought into varieties that grow well in our environment. So we want to be able to take a trait and then transfer it into a well-adapted genotype that's good for here. The problem is when we take that one trait, and we transfer it, sometimes we take extra DNA along with it. And we see a yield reduction, or we see some other issue in quality. So the advantages of using markers or genomic selection is to try to make that selection more precise. So we're only taking over the DNA that we want to take over that is affecting or controlling that trait. So yeah, using these technologies I see in 50 years, the goal would be able to move those trades quicker, faster, and be able to adjust faster to, if it's changes in the environment or changes in, consumer preference as well. So, yeah, we have to take all those things, and being able to be more dynamic, it's a good thing, right? So we got the genetics, and, you'll have the same thing happening on the agronomic side as well. And potentially we'll be able to customize the agronomics for the varieties coming through too. So when farmers get them, they'll say, yeah, this is the rate of nitrogen you should be using, and, everything will be made a little easier so you can, make the most profit off of your variety that you're growing out there.
Mind-boggling to think about, but fascinating.
Yeah. It's not just one thing, right? It's many things, and gradually over time we'll start to see them intersect and be able to work together.
Yeah.
So, yeah.
Thanks for your time, Jennifer.
Yeah.